Hello and welcome to another video which is about the difference between the plasma and serum. Plasma and serum are the fluid which are routinely used in our laboratories, so we should know the difference between these two fluids. The key feature or the key difference between the plasma and serum is that plasma is the fluid which is obtained after the centrifugation of unclotted blood, while the serum is the fluid which is obtained after the centrifugation of clotted blood. Color of plasma is hazy and pale yellow, while the color of serum is clear and straw. We add anticoagulant in the whole blood to prevent the clotting in order to obtain the plasma, while in case of serum we add clot activator, sometime clot activator in the whole blood to stimulate the clotting of the blood in order to obtain the serum. Plasma contains clotting factors while serum does not contain clotting factors because clotting factors has been used in the clotting of blood. Fibrinogen is present in the plasma while fibrinogen is absent in the serum because fibrinogen is the clotting factor and also a protein which has been used in the clotting of blood, so fibrinogen is absent in the serum. When we talk about the isolation time, plasma is less time consuming. It is prepared after when anticoagulant has been added, while in case of serum it is more time consuming and 30 minutes of incubation is required for proper clot formation in order to obtain the serum. Volume of plasma is 55% of the total blood volume, while the volume of serum is less in comparison to plasma. Albumin, globulin, fibrinogen, these three proteins are present in the plasma, while in serum, albumin and globulin are present. Because fibrinogen is the clotting factor and also a protein which has been used in the clotting, so fibrinogen is absent in the serum. So albumin and globulin, these two proteins are present in the serum. Concentration of iron is representative of circulation blood in the plasma, while in case of serum, clot retraction increases the potassium concentration relative to its plasma concentration. This means that in the serum, concentration of potassium is high as compared to its concentration in the plasma or whole blood. Storage condition: frozen plasma can be stored for up to a year, while the serum can be stored at 2 to 6 degrees Celsius for several days. Uses: plasma can be given therapeutically, for example to the trauma patient or the patient with severe liver disease, while the serum is the gold standard for blood banking. And serum is also used in the immunochemistry department for the analysis of many proteins, enzymes, hormones and antigens, etc. So in short or in summary, plasma is the fluid obtained from blood with clotting factors and the serum is the fluid obtained from blood without the clotting factor. Hopefully you like the video. Press the bell icon, comment the topic you want to learn in the future. Stay blessed, stay happy and healthy.